Now we are going to see about history of plant tissue culture. Introduction. Plant tissue culture is the name given to the in vitro method of cultivating cells, organs and tissues, whether animal or plant, with the nutrient solution in stringent lab conditions. A glass container is usually used for this method. History of plant tissue culture. It all began in 1832 when a man by the name of Theodore Scuvan presented the crazy idea that you can grow cells outside of an original host body as stringent external conditions are created. These external conditions would need to create a sterile environment for the tissue culture to develop. 3 years after this seemingly improbable statement, a man named Wilhelm Rox proved the theory in 1835. Rox successfully performed the culture of embryonic chicken cells by using salt solution as his medium. 4 years later, in 1839, and another man by the name of Reichinger proposed the first parameter for this revolutionary method. According to Reichenger's experiment, the tissue culture method would only be successful if the fragments had a minimum thickness of 1.5 mm. Any piece thinner than 1.5 mm would not be able to grow. For almost 50 years after this advancement, the tissue culture method experienced little to no noteworthy mentions. In 1885, a researcher observed the growth of leukocyte cells from salamander in artificial environment, and in 1903, another researcher noticed the same thing. Father of tissue culture. In 1907, Ross Granville Harrison, an American zoologist, was able to culture the nerve cells from a frog in solidified limb. Because of his contributions to the tissue culture method, Harrison now has the title of father. Yeah, he is the father of tissue culture. There were several other successful tissue culture processes throughout the century, and in 1929, the first organ culture was successfully performed in England by a fellow named D.H. Fell. Tissue culture has now been making waves in the plant environment as well. This is mainly thanks to the tissue culture method. It has increased yield, healthier genes, and a sterile environment. Development of plant tissue culture. Although tissue culture has been around since the beginning of the 18th century, plant tissue culture only began developing in 1898. Gottlieb Haberland, a German botanist, made the first attempt to use the in vitro method when grow plant tissues. The cells he used were varied palisade tissues coming from the leaves, the pits, the epidermis, and the epidermal hairs. The initial experiment was fruitful for several months. However, the cells did not proliferate further. Over the years, many experiments used similar parameters. In these experiments, the cells survived for a longer time, but they also failed to proliferate. The first root tips were cultured in 1922, and by making use of subculturing, maintained their cultured roots for 20 weeks. In the 1930s, it was recognized that vitamin Bs and auxin IAA were key components in growing root cultures using the tissue culture method. Conclusion. Over the following century, many similar experiments were conducted and researchers began identifying the most important parameters that sound guide our tissue culture process today. Thank you.